At this point in our discussions, we have focused on the catabolism of glucose and how that breakdown of glucose through glycolysis and ultimately through the citric acid cycle, we can begin to collect electrons with the ultimate goal of producing ATP. What we now need to focus our attention to is how we can use fatty acids as an alternative source to again reach our ultimate goal, which is collecting electrons so that we can ultimately produce ATP. We typically will begin to think of fatty acids and fats as a bad thing in our diet. However, fats are one of the most important things that we can use as a source of potential energy production in our body. We are going to, number one, use sugar, whether that is dietary glucose, whether that is glycogen that we've broken down. That's going to be our primary source of energy. That's the first thing that our bodies go to when we are going to break down. What our second choice is often going to be is fatty acids. Now there are two organs, the heart and the liver, that even during times when we have sugar, we do use the breakdown of fatty acids to provide the majority of our energy needs. But overall, when we're thinking of the human body in total, our number one energy source is going to be glucose. Our number two energy source is going to be breakdown of fatty acids. So we're really going to pull from those fatty acids when we have less sugar available as an energy source. Fats do provide us a very efficient fuel storage for two main reasons. Fats can carry more energy per carbon. What this tip actually means is we have more electrons available per carbon molecule, so we can pull more electrons, forming more reduced electron carriers to ultimately produce energy from our fatty acids. Second advantage of fats is that fatty acids can carry less water. They require less bulk, and so we can store more of them with less weight and things like that. Okay? So the easy way to think of this is for our short-term energy needs, we use glucose and glycogen. When we need energy and that glucose and glycogen is not available or it's been depleted, that's when we're going to move to our longer-term energy storage, which is going to be our fats. We're going to really look at how fatty acids can be broken down to give us the things we need to produce energy. So we need to think about where we can get fatty acids from. Typically, the fatty acids that we will break down for energy are coming from triacylglycerols. If you remember our discussion of fats, you'll remember that there are multiple types of lipids stored in our body. The triacylglycerol is a molecule that is composed of a glycerol backbone with 3 attached fatty acids. The main function of a triacylglycerol is for energy storage. We had other types of lipids such as phospholipids that mainly produce our membranes. We have sterols. Those other lipids can be broken down, and there are ways we break them down in our body, but their main purpose is not to be broken down for the production of energy. So we're really going to think mainly about our triacylglycerols as we go through fatty acid catabolism. Triacylglycerols can come from a dietary source, and we're going to look at exactly how we break down those triacylglycerols in our diet. The triacylglycerols can be broken down from immediate dietary intake, or those triacylglycerols from our diet can be stored for later. Once the triacylglycerides are stored for later in our adipose tissue, we can then break those down as a source for fatty acids to break down. Fats that are synthesized in our body can be broken down, but that's going to be kind of the third option when we're thinking about sources of fatty acids in our diet, in our body for energy use. This figure shows us kind of an overview of how we can take these dietary fatty acids and break them down and get them where we need them to be free fatty acids that we can oxidize for fuel. So we're going to ingest fats in our diet. Those fats will go through our digestive tract. When the fats get to our small intestine, bile is released from our gallbladder to properly emulsify our triacylglycerols. Once emulsified, a lipase is going to be able to break down the triacylglycerol, releasing our fatty acids as well as our glycerol backbone. The released fatty acids are going to be incorporated into molecules for transport called chylomicrons. The chylomicron is going to be a shuttle unit The inside of the chylomicron, as you can see here, is composed of basically a big ball of triacylglycerol with some cholesterol esters. The outside of our chylomicron has some proteins called apolipoproteins that are going to be important for directing where that fat needs to go in the body. So it's going to be kind of the little signaling flags. A particularly important one for our discussions now is this apolipoprotein C2. C2 is going to be the trigger when this chylomicron gets to an area where we need to release these triacylglycerols. That's going to be the signal. So if we go back to our same picture, we're here at this point, it's labeled number 4 on this picture. We've packaged our dietary triacylglycerols. We then move them through the bloodstream until that apolipoprotein C2 recognizes an area and another lipase is activated. The activated lipase will again break down the triacylglycerol, releasing the fatty acid into the cell. In that particular cell, that fatty acid can then be broken down through a process called beta oxidation. So as you can see from that short discussion, we can take dietary lipids, break them down into free fatty acids, and that can be used for production of energy. We also can take the triacylglycerols that we have taken and put into our adipose tissue, and when we release glucagon and epinephrine, that can be a signal of it's time to break down those stored triacylglycerols. So if we look here at this picture, when we release glucagon, remember glucagon is the signal for I don't have any sugar, I need something else as an energy source. We take the glucagon, binds a G protein coupled receptor, which in turn activates adenylyl cyclase to convert ATP into cyclic AMP. Cyclic AMP then activates protein kinase A. Protein kinase A is now going to phosphorylate 2 different things. Protein kinase A is going to remove the inhibition we have with this perilipin molecule, and protein kinase A is going to phosphorylate our hormone-sensitive lipase, activating it. The lipase will go into our fat cell, our adipocyte. The lipase breaks down stored triacylglycerol, releasing fatty acids. Fatty acids move in the bloodstream, combined with some serum proteins, and moves to particular areas and cells where we need to break down that fatty acid through beta oxidation. We don't waste anything. We don't only break down the fatty acid, we're also going to break down the glycerol that's released from the triacylglycerol. So one enzyme located in the cytosol is going to take released glycerol, add a phosphate, forming glycerol 3-phosphate. Following this initial reaction, glycerol 3-phosphate dehydrogenase is then going to convert the glycerol 3-phosphate into dihydroxyacetone phosphate. Dihydroxyacetone phosphate is an intermediate in the glycolytic cycle. It can then enter the normal glycolysis pathway, convert into glyceraldehyde 3-phosphate, and then continue into the breakdown process just as we would if this glyceraldehyde 3-phosphate originated from a breakdown of glucose. All right, so this is what we're going to do with our glycerol we release. What do we do with the fat that we release, the fatty acid? How do we break that down? The first thing we're going to do with the breakdown of our fatty acid is activate the fatty acid so it can then be properly moved into the mitochondria. All of the reactions of glycolysis happen in the cytosol. At this point, the fat is in the cytosol. We've moved it through the bloodstream into a cell where the fat is needed to be broken down. Now our fat needs to go from the cytosol into the mitochondria for beta oxidation. To get our fat into the mitochondria, we take our fatty acyl group here. The fatty acid is then combines with the adenosine and 1-phosphate from an ATP molecule. So 2 phosphates are cleaved, we get a pyrophosphate, and we get the adenosine, 1-phosphate, attached to the fatty acid, which is represented here by this pink molecule. In the second part of the reaction, a CoA molecule comes in and we form a high-energy thioester bond, releasing the AMP. Ultimately, we get the fatty acid, the R group, attached through a thioester bond to a CoA. So this is called fatty acyl-CoA. A driving force for this reaction is we will also take this pyrophosphate that was released from the beginning reaction, break it into two inorganic phosphates. Two enzymes make all of this on this page happen. We have the fatty acyl-CoA synthetase that has a two-part reaction. The fatty acyl-CoA synthetase attached the fatty acid to the AMP and then coordinated the attachment of the CoA to the fatty acid. A separate enzyme, the pyrophosphatase, is going to cleave this pyrophosphate, giving us some more free energy to drive the reaction. We now have our fatty acyl-CoA ready for transport into the mitochondria. Transport into the mitochondria requires the molecule carnitine. Once the carnitine assists with the transport of the fatty acid into the mitochondria, we can then complete beta oxidation inside of the mitochondria. You may recall that the mitochondria has two membranes. It has an outer membrane as well as an inner membrane. There is an enzyme in the outer mitochondrial membrane called carnitine acyltransferase 1. There's also an enzyme near the inner mitochondrial membrane called carnitine acyltransferase 2. The fatty acyl-CoA that we've just produced from this particular reaction here, from the fatty acyl-CoA synthetase, that fatty acyl-CoA combines with carnitine and is transported across the outer mitochondrial membrane through the inner membrane space and into the matrix. So here we have fatty acyl-CoA attached to carnitine. We're then going to take the carnitine off. Carnitine can be recycled back out to the outer membrane for use again, and we release a fatty acyl-CoA into the matrix. So you can see all we've done is transport exactly what was in the cytosol here into the mitochondrial matrix so we can complete beta oxidation. The carnitine is never used in the reaction. It is simply a little transporter that carries the fatty acyl-CoA into the mitochondrial matrix. Now that the fatty acid is inside of the mitochondrial matrix, we can complete the breakdown of fatty acid, which is called beta oxidation. And that's shown here in stage 1. So beta oxidation is going to take this long chain fatty acid, which is a long chain of carbons, and break it down into 2 carbon units, releasing acetyl-CoA. So you can see those acetyl-CoAs that we release from breaking 2 carbons off of the fatty acid can enter the citric acid cycle. So let's look specifically at the reactions that are going to break off that acetyl-CoA from the fatty acid. Beta oxidation has 4 steps. In step number 1, we remove an electron and form a double bond. The initial enzyme, acyl-CoA dehydrogenase, takes our long-chain fatty acid, begins to oxidize the molecule, so we release an FADH2, and we ultimately end with this trans double bond. It's important to note that this is a trans double bond because that's unusual in these long-chain fatty acids. Recall from our discussions of fatty acids, when we do have an unsaturated fatty acid, the double bond is always in the cis conformation. The second enzyme of the beta oxidation pathway is going to work specifically on a trans double bond. It is a hydration reaction, hence the term hydratase. We add a water molecule, converting this trans double bond into an alcohol group. Then in step 3, we have our next dehydrogenase, again further oxidizing our fatty acid chain, producing NADH. From this, our last step, we are going to be able to break off our first 2 carbons, we release the first two carbons with the CoA attached, so we bring another coenzyme A in, so that we end up with a fatty acid chain that looks very similar to the beginning fatty acid chain. It's got the CoA on the end. The only difference is it is 2 carbons shorter than the beginning. This released acetyl-CoA can now go into the citric acid cycle. So you'll notice that for each cycle of beta oxidation, we produce 1 FADH2 and 1 NADH. So if we go back to this picture, if in this picture we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 carbons, we will have to do 7 cycles of beta oxidation. So it means we're going to cut 2 carbons off 7 different times. So we'll have 7 cycles of all 4 reactions. We will produce 7 NADH molecules and 7 FADH2 molecules from the beta oxidation pathway. In addition to the 7 NADH molecules and the 7 FADH2 molecules, we will release 8 acetyl-CoAs. Those 8 acetyl-CoAs will each go through the citric acid cycle. So we'll produce 8 times 3, which is 24 NADH molecules from citric acid cycle. We'll produce 8 times 1, which is 8 FADH2 molecules from the citric acid cycle. And we'll also produce 8 times 1, which is 8 GTPs. That's equivalent to 8 ATPs. For study purposes, here's your steps of the citric acid of beta oxidation. Overall energy yield shown here as I was just explaining, and the actual math. In class, I gave you the assignment of figuring out the energy yield for a 20 carbon fatty acid. I highly recommend that you practice with this using different length fatty acids to make sure you understand the overall energy yield. This is the energy yield for the example shown in the picture. 16 carbon long fatty acid. So you can see from this, from the 16 carbon long fatty acid, we will ultimately produce 108 ATP molecules. From 1 glucose, we produced 32 ATP molecules. So we do get more energy from breaking down fats. Not all fatty acids in the body are saturated fatty acids, as we've seen from that initial discussion of beta oxidation. We do also have some unsaturated fatty acids. We have polyunsaturated fatty acids, and we have monounsaturated fatty acids. So we'll need some extra enzymes. We'll need an isomerase because the double bonds in our unsaturated fatty acids are in the cis conformation, and we need to change them to trans double bonds so they can enter into the beta oxidation pathway. We also will need reductase to further reduce some of the cis double bonds. If we have a monounsaturated fatty acid, we only need isomerase. We just need to change the cis to a trans. If we have polyunsaturated fatty acids, we'll need the isomerase as well as the reductase. So I want to show you some example reactions of how this would look. These are not reactions that you need to memorize. You just need to understand how it will affect energy yield and the steps required of beta oxidation if we do have an unsaturated fatty acid. So in this example, this is a monounsaturated fatty acid shown here at carbon number 9. So what we would do is we would do normal beta oxidation, step 1, step 2, step 3. So we go through 3 cycles of normal beta oxidation until we get to the double bond. Once we get to the double bond, we will just need this isomerase. It will change it to a trans fatty acid. Now we can enter directly into step 2 of beta oxidation. Keep going for 5 more cycles of beta oxidation. This is not going to affect the energy yield at all. The isomerase reaction does not require any energy. With a polyunsaturated fatty acid, we would continue the normal steps of beta oxidation until we get to a double bond. Then the isomerase is going to make it trans, finish beta oxidation. Then we would get to another double bond. What we would need to do is use the reductase enzyme to get the second double bond and trans conformation. Then we can continue through beta oxidation as normal. Just to make sure our discussions are complete, let's think about what would happen if we had an odd-numbered fatty acid. An odd-numbered fatty acid, we would continue with normal beta oxidation pathways until we ended with that last 3-carbon fatty acid. The last 3-carbon fatty acid would give us this propionyl-CoA. Propionyl-CoA will then continue through a few steps until we ultimately produce succinyl-CoA. Succinyl-CoA can then enter into the citric acid cycle so that we can actually continue using these molecules. Remember that we never want to think of this as anything can be wasted. We're going to incorporate it and move it into reaction somehow. Again, these are not enzymes that you need to memorize or pathways you need to memorize, but you should understand that if we do end up with a odd carbon fatty acid from breaking down plant fats or something of that nature, then we would take that 3 carbons, convert it into succinyl-CoA, and enter it into the pathway. Regulation of fatty acid synthesis is can be considered a little bit convoluted. We need to think about a couple of enzymes that we hadn't really talked about yet, but let's think about what is going to trigger whether or not we want to do fatty acid breakdown or not. If we're doing fatty acid breakdown, then we are not going to be doing fatty acid synthesis. So you kind of think of them as opposites, okay? So if we have a lot of sugar available, a lot of sugar triggers the release of insulin, right? Insulin is our hormone that tells us, hey, there's a lot of glucose available. If we have a lot of glucose available, we've already learned that insulin is going to trigger glycogen synthesis. We're going to store that excess glucose. Insulin is also going to trigger fatty acid synthesis. If we want fatty acid synthesis to occur, then we're going to need to block the movement of fatty acids into the mitochondria. If a fatty acid goes into the mitochondria, we're breaking it down. So insulin is going to make our enzyme here, this ACC enzyme that's responsible for taking acetyl-CoA and carboxylating it. This is the first step of fatty acid synthesis. So when insulin's available, we activate fatty acid synthesis, producing a compound called malonyl-CoA. Malonyl-CoA comes over, blocks the transport of fatty acids into the mitochondria. If we're making fatty acids, we don't want to waste time making them if we're just going to move them in and break them down. Glucagon is going to have the reverse effect. Glucagon tells us that we have low blood sugar. Low blood sugar means break down glycogen storage, give me some sugar, but also begin breaking down fatty acids. If we are breaking down fatty acids, then we need our carnitine acyltransferase active. Okay? So glucagon is going to make the enzyme that makes fat inactive. If we are not making fat, then we are not making malonyl-CoA. If malonyl-CoA is not available, then it's not inhibiting our enzyme. So all of our fatty acids available will go into the mitochondria and we will begin beta oxidation. So the easiest way for me to remember this is insulin triggers sugars available, don't break down fat, make fat. Glucagon triggers, I don't have any sugar. I need something. So let's break down my glycogen. Let's break down my fatty acids. The last thing that we need to discuss in this chapter is related to fatty acid breakdown, and that is the formation of ketone bodies. Anytime we have large amounts of acetyl-CoA, they can go into the citric acid cycle, which is what we've been discussing. So if we've done a lot of glycolysis, or if we've done a lot of fatty acid breakdown and we have large amounts of acetyl-CoA, those can go into the citric acid cycle. Sometimes the large amounts of acetyl-CoA that we produce will get the citric acid cycle to the point that we've ran out of oxaloacetate. With no oxaloacetate, there's nothing to combine with acetyl-CoA, so the citric acid cycle can't continue. In that instance, acetyl-CoAs will then go into what we call ketone body formation. Ketone body formation looks like this. Two acetyl-CoA molecules are combined together through the enzyme thiolase. We form acetoacetyl-CoA. Acetoacetyl-CoA will then combine with a third acetyl-CoA molecule, forming this HMG-CoA, beta-hydroxy-beta-methylglutaryl-CoA. Long, horrible name. Look at what we're doing. That's more important than memorizing structures and names. We basically took 3 acetyl-CoA molecules and we stuck them together to form a larger ketone body. Remember, we're only going to do ketone body formation if we have a large amount of acetyl-CoA. So if we have a large amount of acetyl-CoA, we just start putting them all together. Once we make this HMG-CoA, we can then continue with a few other reactions, making acetoacetate. Acetoacetate can then be used to make acetone or this beta-hydroxybutyrate. Beta-hydroxybutyrate is actually our largest concentration ketone body that we have floating around in our bloodstream. Okay? So let's think about what are conditions that would lead to high formations of ketone bodies. The two most common that we think about are starvation as well as diabetes. So let's think about starvation at first. If we are starving, that means we are not eating, we have no sugar available. If there's no sugar available, we've already broken down all of our stored glycogen. We're to the point that we need something. Our brain specifically needs sugar, and if we're starving, there is no sugar available. Your brain can live off of ketone bodies when it's necessary. Okay? So no sugar available, what are we going to start doing? No sugar is available, we're going to increase gluconeogenesis. Gluconeogenesis uses oxaloacetate. If we're starving and we're trying to make sugar, we've pulled our oxaloacetate away through gluconeogenesis. If we're starving, we're going to break down fats. Breaking down fats releases what? Acetyl-CoA. We'll now have high levels of acetyl-CoA and not enough oxaloacetate to go through the citric acid cycle. So those large amounts of acetyl-CoA will activate ketone body synthesis in the liver. The liver then releases those ketone bodies into the bloodstream. Ketone bodies can go where they're needed, specifically the brain. Acidosis is a very common side effect where the blood pH is going to get very low in these situations because we're releasing all of these acids into the bloodstream. So how does this relate to diabetes? With certain types of diabetes, your body may have sugar, but it's going to mimic the effect of starving because your body doesn't realize it has any sugars that can be used. So with diabetes, a lot of times we will mimic starvation. Our bodies will begin producing these ketone bodies. So we've made ketone bodies. We're starving. These ketone bodies go to the brain. What does the brain do with them? The brain will then catabolize the ketone bodies. It's going to break down that beta-hydroxybutyrate, releasing NADH, releasing succinate, ultimately releasing acetyl-CoAs. Hopefully, in the brain, we will then be able to use the acetyl-CoA, use the succinate to enter the citric acid cycle so we can continue producing energy for that organ. So why will the brain have oxaloacetate so that citric acid cycle can continue if the liver did not have oxaloacetate? Remember, the liver was using the oxaloacetate available to do gluconeogenesis. The brain can't do gluconeogenesis. So two different things are occurring in two different parts of the body. What we're seeing in the liver, so this is a liver cell, the liver is taking fats and breaking them down to try to release an energy source. That acetyl-CoA can't go through the citric acid cycle because the oxaloacetate has been converted into glucose so that the glucose can go to the brain. All the excess acetyl-CoAs make ketone bodies. So the ketone bodies and the glucose that the liver makes can go other places, enter the citric acid cycle, producing reduced electron carriers, and ultimately producing energy.